Earlier, we have discussed how histone tails can be post-translationally modified to alter chromatin compaction and access to the DNA. This video provides an introduction to the molecules that read histone acetylation. As we have discussed previously for other marks, the mechanisms governing epigenetic marks such as histone acetylation can be summarized by the writer, eraser, and reader model. For histone acetylation, the writer proteins are histone acetyltransferases, the eraser molecules are histone deacetylases, and the reader molecules are bromodomain-containing proteins. Here, we will focus on the readers of acetylation marks. The major reader domain for acetylation marks is the bromodomain. The bromodomain is a conserved protein domain of approximately 110 amino acids. In the figure to the right, you can see the molecular structure of a bromodomain. It consists of 4 alpha helices connected by variable loops. Both the ribbon diagram on the left and the space-filling diagram on the right include the histone tail peptide that is the target of this bromodomain in yellow. In the case of the PCAF bromodomain that is shown here, the target is H3 lysine 36 acetylation. The target specificity of the bromodomain is determined by small differences in the bromodomain itself, as well as in adjacent amino acid residues. Often, bromodomain proteins contain other chromatin binding domains that also contribute to the binding specificity of the protein. Most eukaryotes have a large number of different bromodomain proteins that interact with both acetylated histones and other acetylated proteins. This diversity in bromodomain proteins is illustrated here for humans, which have 61 bromodomain-containing proteins. These proteins are classified into 8 groups depending on their amino acid sequence similarity. Bromodomain proteins carry out a variety of functions in the cell. They tend to occur in complexes and, similar to the histone acetyltransferases, sometimes act on non-histone proteins. The bromodomain proteins are often involved in chromatin remodeling, where they help with the decompaction of chromatin and promote gene expression. They also have been linked to cancer and inflammation. Because of their link to various diseases, bromodomain proteins have also been the subject of drug trials. Inhibiting the binding of a bromodomain protein would be expected to have a similar effect as the inhibition of the HAT protein. If there is no reader protein to initiate a response to an epigenetic mark, it is similar to the absence of the mark. Several bromodomain inhibitors have been identified, and they are currently used in clinical trials for a variety of cancers, diabetes, and atherosclerosis. Up until approximately 5 years ago, the bromodomain was the only known reader of histone acetylation. Recently, there have been new reports of 3 additional reader molecules for histone acetylation. A PhD domain has been found to recognize acetylated H3. A tandem pH domain from the yeast RTT106 protein can recognize acetylated histones, and the yeast domain of human ASF9 can recognize several acetylation sites on H3. In summary, bromodomain proteins are the most common readers of histone acetylation. Their specificity is determined by sequence variation within the bromodomain, as well as adjacent residues and protein domains. Bromodomain proteins often function as a part of complexes and help with chromatin decompaction. Due to their role in decompaction and gene activation, bromodomain proteins are drug targets, and several bromodomain inhibitors are in clinical trials. New acetylation reader domains have been identified recently, but their number is small compared to the bromodomain proteins.